Excellent. Well, thank you, Kevin. I really appreciate that introduction, the kind introduction. And as Kevin said, I'm an assistant professor here in Sky School of Pharmacy at the CU Anschutz campus. And if you look at this picture, well, I guess it's on the other side, just on the other side of the glass and steel outcropping there. My office is just on the other side of that, at about that, at that second level there. So today I'm going to talk to you about using LCMS to identify CYP3A7 drug hormone interactions, and I modified the title a little bit because I'm also going to talk about how they affect toxicity of drugs in the neonate. So my lab is a drug metabolism pharmacokinetics lab. So we're doing a lot of small molecule metabolite identification and quantification. But before I start, as I used to say, we'll have a word from our sponsor. And in this case, it's water. So this is for you, Steve. So we bought this instrument, I guess, late last year. Is that right, Steve? The Zevo TQS Micro.
Yeah,
So
I think it was the middle of the year. Thanks, Jeff.
Has it been about a year? Yeah. Well, it's a great instrument. So a lot of the work we do is more in metabolite quantification than metabolite identification. So sensitivity was really important to us. And of course, being an academic lab, cost was important as well. And so Steve cut us a great deal on the TQS Micro. And it's been a really nice instrument. We just had the PM on it last week. Probably really didn't need it because it hadn't got as much use as we would have liked during COVID-19. But it's a great instrument for what we're doing in the lab. And just to give you an idea of where it fits in with the other triple quad instruments that Waters sells, it's sort of a mid-range instrument. So sensitivity is really important to us, but of course, we have to balance that with the cost. And in this case, the TQS Micro is great because it has a small footprint. It's pretty sensitive. It has this radar acquisition mode. We haven't really used that yet, but that can be efficient in identifying some of these metabolites. We have two different, we have both the ESI probe and the APCI probe for quantifying different types of metabolites, including endogenous metabolites such as steroids. So that's been really, really handy as well. So it's definitely a nice instrument and I can recommend it. But what I really want to talk to you about today is babies, right? Babies, yes, you can go, ooh, ah, everybody loves babies. We love this one. This is our little baby, though she's not little anymore. She's like 10. So she keeps saying, Daddy, don't show that picture. I have a newer picture. But we love babies, of course. But the problem is, in this country, we dose babies with drugs all the time in the NICU. They've never been tested for safety and efficacy on this particular population. And we make the assumptions that basically the drugs are going to act exactly as they do in the adult if we just scale down the dose. And that may not always be the case. And I'm going to show you a couple of examples where it's not and why it's not. And in fact, the off-label use of medications, because keep in mind, no clinical trials are done on babies, right? So the physicians are prescribing these drugs off-label. And the use of off-label medications is really increasing in pediatric patients. A recent meta-study found that about 10% of patients in the NICU received at least one off-label drug, and in many cases, more than one drug. And that's pretty typical, right? Because obviously the baby's very sick to end up in the NICU to begin with, and they're going to be prescribed with multiple drugs right from the get-go, right? Usually what happens is the babies get a lot of medications right after birth, and then that's sort of titered off over time. And so more than half of these patients obviously are newborns, and the use of multiple off-label drugs then increases the risk for drug-drug interaction. So what do I mean by drug-drug interaction? So this is an interaction that occurs when you have one drug that's dosed simultaneously with a second drug, and that second drug affects the levels of the first drug, either through a pharmacokinetic interaction, so for inhibiting its metabolism, so you either get increased concentration of the drug, or activating its metabolism, so you get a decrease, or sometimes through pharmacodynamics, where it inhibits binding to the receptor that's important for its action. And the American Academy of Pediatrics is predicting that off-label use of medication is only going to increase in the future. And there's a very good reason for this because, again, drugs aren't, designed, they don't design clinical trials to look at drug effects in neonates. And so they have to prescribe them off-label. They don't have much of a choice. But this really begs the question, if babies are just like little adults. I mean, we know a lot about drug metabolism in adults, but how well does that correspond to what's happening in babies? I'm sorry if you hear a little music in the background. My daughter's apparently doing her piano lesson. In any case, the question becomes, can we use what we know about metabolism in adults and extrapolate down to babies? And essentially, this is what's done, right? So physicians use allometric scaling. So essentially, they take into account height, weight, body surface area, and then just scale down the dose based on that. But they're assuming everything else is the same, including metabolism of the drug. And in fact, it's not. So what I'm showing you here is a chart comparing 3 cytochrome P450 enzymes. So you might remember cytochrome P450s are the main class of drug metabolizing enzymes in your liver. So drugs get cleared mainly through the liver. These are oxidative enzymes, so they're attaching a hydroxyl or epoxide to the drug, making it more water soluble so it can be excreted in the urine or the bile. And there's several of these P450s in your liver, but the most important one is one called CYP3A4, cytochrome P450 3A4. And that enzyme, single enzyme, catalyzes the metabolism of about 50 to 60% of all of the drugs on the market. Analogous enzyme CYP3A5, which is present in certain ethnic populations, also catalyze that reaction, but it's much less prevalent. In infants and neonates, you have an enzyme called CYP3A7. Now, as it turns out, infants and neonates don't really express much of this 3A4 enzyme. They express all of this instead they express 3A7. So if you look here on the graph, and so what you're looking at is they used Western blot to quantitate the 3 different CYP3A isoforms. And before birth, so this is the estimated gestational age is what the EGA is referring to. Before birth, you see very high levels of CYP3A7, almost no 3A4 and 3A5. This begins to decline until to about 6 months of age. And then you start to see CYP3A4 take over. So this can have profound implications in drug metabolism and drug disposition in this particular population because you have a completely different type of enzyme there. So how different are these enzymes? Well, if you compare sequence identity, they're pretty similar. It's about 87% sequence identity between CYP3A4 and CYP3A7. And in fact, although we're working on a crystal structure, at least my collaborator at Michigan's working on a crystal structure for 3A7, we don't have it yet. But we have every reason to expect it's going to take a very similar fold to CYP3A4. These are heme-containing enzymes, so they have to be able to bind that prosthetic heme group in a certain fashion. And all P450 folds look more or less the same. So structurally pretty similar. Substrate range, well, again, CYP3A4 metabolizes 50 to 60% of the known drugs in the market. And it looks like so far CYP3A7 does as well. And the substrate specificity between the two overlaps quite significantly. The average KCAT, though, if you compare that with 3A4 and 3A7, for your average drug substrate, 3A4 catalyzes about 10 to 100 turnovers per second. Now compare that with CYP3A7. CYP3A7 is more like 0.1 to 1 per second. So it's about 100-fold less active on the average drug substrate. What is that going to do to drug clearance? Obviously, most drugs are going to get cleared a lot slower in the infant liver than they are in the adult, just because this enzyme is much slower. In terms of regiospecificity, so where those drugs get oxidized, that varies from CYP3A4. You can have multiple oxidations occurring at different positions on the drug. Same thing with CYP3A7, although it might not always be identical. I'm going to show you some examples of that, what happens with that. Endogenous substrates, CYP3A4 supposedly metabolizes testosterone, although there's some debate about it. It does metabolize it, but whether it's an endogenous substrate, there's some debate about that. In terms of endogenous substrate for CYP3A7, there's two important hormones that get oxidized by CYP3A7, dihydroxyepiandrosterone sulfate, which is the precursor to all the downstream steroids, both the androgens and estrogens, and also retinoic acid. So both of these compounds are really important for growth and development in the fetus and the neonate. And then finally, there's some ancillary effects with cytochrome B5. So this is another protein that's in the liver. It enhances the reaction in most cases for 3A4. It doesn't seem to have any effect on 3A7. So the enzymes are pretty different functionally. Now, a little bit more about what 3A7 is actually doing in the fetus and neonate. This is to differentiate it from 3A4. 3A4 you can think about as like the garbage cleaner of the liver, right? All these drugs and xenobiotics are coming into the liver. You got to get rid of them because they're essentially going to build up the toxic levels if you can't. So 3A4 comes along, hydroxylates them, they become more water soluble, and you can excrete them. 3A7, on the other hand, has a completely different type of function, an endogenous function. Remember, a lot of these P450s also metabolize steroids and are important in synthesis of steroids. And in 3A7's case, it hydroxylates DHEAS to 16-hydroxyl DHEAS, which gets exported from the fetal liver into the placenta and eventually converted into estriol. So why is this important? Well, it turns out estriol is really critical for fetal growth and development during that last trimester of pregnancy. And in fact, it's the highest circulating estrogen during that last trimester of pregnancy. And women that have extremely low levels of estriol tend to give birth prematurely to low birth weight infants. So it seems like it's a signal from the fetus to the placenta and to the mother that everything's going well. You should continue on with the pregnancy and until the full term. So anything that could inhibit this and reduce those 16-hydroxy DHEAS levels will lead to reduced estriol and birth and low birth weight, which is the primary cause of infant mortality in the US. So keep that in the back of your mind. So drug metabolism then impacts both safety and efficacy, right? So safety, and I'm going to tell you two stories today. One is about safety, and the other's about efficacy of the drug. And so, how does it impact these two things? Well, it impacts safety when a drug is metabolized too slowly. It can build up in the bloodstream, and then toxicity can result. Also, similarly, if a reactive metabolite is produced when that drug is metabolized, hepatotoxicity can occur or other types of toxicity. And a classic example of this is the metabolism of acetaminophen to a reactive metabolite called NAPQI, which is the quinonamine, which reacts with proteins in the liver, and this results in liver toxicity. It can also affect efficacy if a drug's metabolized too quickly. Now, that's probably less the case with 3A7, but when that happens, it may never actually reach the effective concentrations at the site of the receptor. And this can decrease or eliminate the therapeutic effect. An example of that's with HIV protease inhibitors, ritonavir, which we'll talk about in the second part today. So the first story then is about 3A7 and a drug called nevirapine. So nevirapine is a non-nucleoside HIV reverse transcriptase inhibitor. It's been used clinically for over almost 25 years now, something 6 years. And it's known to cause hepatotoxicity in about 6% of the patients that use it, but otherwise it's a pretty safe drug. It's also been reported to cause hepatotoxicity in neonates. So that generated our interest in it. And the fact that it's the primary drug given to HIV-infected mothers and their infants at birth. Certainly, this is pretty prevalent in the third world, where nevirapine's off patent now. So it's one of the cheaper and more effective anti-HIV agents. So it's typically given to pregnant women and their infants. In terms of metabolism, nevirapine metabolism in adult human liver microsomes is complex. And there are a lot of P450s involved in it, including CYP3A4 and CYP2D6. Those are the primary P450s. And this is pretty typical with drugs, right? That they'll get metabolized not just by 3A4, but some of the other P450s as well. although it turns out 3A4 is the major isoform that metabolizes nevirapine. And the majority of the metabolism generates 2-hydroxynevirapine and also 12-hydroxynevirapine, which can also be converted into this 4-carboxy species. There's some other minor metabolites, but those are the major metabolites that are formed. You can see that here. Whoops, I'm sorry. You can see that here as well. If you look in the left, and so what they've done here is they're looking at recombinant purified protein, the CYP2B6, 2D6, 3A4, and 3A5 to see which one produces which metabolites and how much. It's called reaction phenotyping, pretty typical thing that's done in the drug industry. You can see with 3A4 here, it produces both the 2-hydroxy and this 12-hydroxynevirapine. Now, in addition to those metabolites, it turns out CYP3A4 also produces a reactive metabolite. And in fact, if you do the incubation in the presence of GSH, which acts as a nucleophilic trapping agent, you can see that most of this reactive metabolite is produced by CYP3A4, although CYP2D6 contributes a minor portion to production of the reactive metabolite. As it turns out, that reactive metabolite can also adduct nucleophilic residues in the enzyme, leading to enzyme inactivation. And so if you look here on the left, what I'm showing you here in this chart is we're analyzing the percent remaining enzyme activity after we've done an incubation with nevirapine. So typically how these are done is you take your drug that you think generates reactive metabolite, in this case nevirapine, you do what's called a pre-incubation with that for 10 or 15 minutes, and then you take an aliquot out and do a secondary incubation with another drug that's not doesn't generate reactive metabolites, but it acts as your reporter, and you quantify metabolism of that drug to understand how much remaining activity you have left. And what you can see here is we're looking at a function of time and increasing concentration from the top to the bottom. So this is, I'm sorry, it's not marked. This is 0 micromolar nevirapine, 2 micromolar, 5, 10, 15, 20, and 100. You can see with increasing concentration of drug and an increasing amount of time, you see more inactivation of the enzyme. And this is very characteristic of time-dependent or mechanism-based inactivation of an enzyme, suggesting the production of reactive metabolite. You can also take the slopes and fit them to a binding isotherm and retrieve your Ki of, in this case, 31 micromolar and Kinact of 0.029 per minute. So there's less enzyme activity as a function of time when you incubate with nevirapine, suggesting you have production of reactive metabolites inhibiting the enzyme. And in fact, what happens is the parent compound, nevirapine here, again, gets metabolized by CYP3A4 in the adult and 2D6 to generate this unstable intermediate, which can recombine, that radical can recombine with hydroxyl to form the 12-hydroxynevirapine and the 4-carboxy, which gets further metabolized to the 4-carboxy. And both of those are inactive. However, it can abstract a hydrogen radical to generate this quinone methide species, which has this reactive electrophile here, which can then react with nucleophiles in the cell and generate the hepatotoxicity that we've seen. And this has really been elegantly demonstrated in rat models. So we know that this reactive metabolite is responsible for the hepatotoxicity that we see in the adult. So our hypothesis then was that CYP3A7 will metabolize nevirapine to produce a qualitatively similar metabolic profile seen with adult CYP3A4. So we expect to see similar metabolites, 2-hydroxy, 12-hydroxy, and the reactive metabolite, albeit we also expect to see important quantitative differences. You might expect that you would produce fewer amounts of metabolites than you would with 3A4. And it turns out the significance of this is that increased production of this quinone methide species, which was counterintuitive to what we thought initially, by CYP3A7 for metabolism of nevirapine can put neonates at higher risk of liver injury because you're producing this reactive metabolite with CYP3A7 more so than CYP3A4. So here's, I'm going to show you guys some LCMS now. So first, we did some incubations with nevirapine, just with the recombinant enzyme. So this is recombinant purified CYP3A7. And what I'm showing you here is production of, we could show we produce both the 2-hydroxy species, the 12-hydroxy, and the GSH adduct. Now, how do we know these are what we think they are? I'm just going to show you in a second. The next slide, we have authentic standards that we were able to purchase from Toronto Research Chemical. So we were pretty confident of our identification and quantification of the different metabolites. Now, a pretty typical experiment that's done when you're doing these incubations, either with the recombinant enzyme or human liver microsomes, is a plus and minus NADPH experiment. So NADPH donates electrons to cytochrome P450 reductase, which is the reductant for the P450. So it eventually transfers electrons directly to P450. So if you don't have NADPH present, you're much less likely to see any sort of metabolism. So this is kind of a good check to make sure you're seeing metabolism specifically from the enzyme, and it's not some background effect. And in fact, what we see here when we add in NADPH, we can identify that 2-hydroxy nevirapine species, the GSH adduct, and the 12-hydroxy nevirapine. So very similar profile to what we saw with 3A4, with 3A7, although we're not making as much metabolite with 3A7, at least for the 2-hydroxy and 12-hydroxy. So what I'm showing you here is just the standards that we use to do the quantification of the 2-hydroxy nevirapine using this synthetic standard. We did some MSMS to make sure the fragmentation patterns fit what they did with the standard. So we were able to confirm that 12.8-minute peak was 2-hydroxy nevirapine. Similarly, we did an experiment with the 12-hydroxy standard and confirmed that the 14.2-minute peak was the 12-hydroxy. Now, the reactive metabolite was more of a challenge, right? Because we can't just buy, well, this GSH adduct standard, right? So we weren't able to quantitate it specifically with LCMS, but we were able to look at the fragmentation pattern and also and identify it that way, and also by identification of that parent peak, the parent ion. So we were able to confirm that peak at 13.9 minutes was in fact the GSH adduct from that reactive metabolite, which you see the structure shown there. Okay, great. So we have a similar metabolic profile with 3A7, the purified enzyme, to 3A4, exactly what we were predicting. But what about in a more realistic system, right? So sometimes you can see metabolism with purified enzyme. It might not really be reflective of what's happening in the liver. Now, unfortunately, or fortunately, we can't take people's livers and just do metabolic studies on them. So we have to rely on donor livers. And so from donor livers, we can make human liver microsomes. And in fact, we had a donor liver from a 6-month-old that we were able to make human liver microsomes from and do the incubation of nevirapine human liver microsomes. And we were able to identify the 2-hydroxy species, as well as the 12-hydroxy, and again, the GSH adduct. Although you'll see that the peaks are much less intense here, indicating, we're making a lot less metabolite than we did with the purified enzyme. Not a huge surprise. It's probably due to the fact that we have lower levels of 3A7 in the human liver microsomes than we did, when we were doing the purified incubation. So to try to understand then if we're making the same amounts of these metabolites, we turned to doing some kinetics analysis since we had a nice, good analytical assay in hand. And so initially we looked at the 2-hydroxy species. So this is the inactive metabolite. This is a good metabolite. It helps us clear the drug. We want to get rid of it. And we compared formation of this metabolite with 3A4, recombinant 3A4 versus 3A7. And you can see, so the Km for 3A4 on the left is 61 micromolar. The Km for 3A7 is 95 micromolar, fairly similar, within 2-fold. That's good enough for as a comparison with P450 enzymes. But you can see the Vmax between the two is significantly different, almost the 20-fold difference between 3A4 and 3A7, with 3A4 being 0.317 picomoles per minute per picomole, and the Vmax for 3A7 being 0.1 0.018. So again, what we would have predicted, right? 3A7 is a crappier enzyme than 3A4. It doesn't metabolize drugs quite as well as, not anywhere near as well as 3A4. We also compared kinetics of the 12-hydroxy species formation. And this was a little bit of a surprise because although the KMs are still within twofold here, 73 micromolar for 3A4 versus 114 micromolar for 3A7, the Vmax was a lot closer than what we expected to see. So it's the Vmax for 3A7 for the 12-hydroxy formation is only threefold less than that for 3A4. And in fact, if you compare the ratios of those two metabolites, so the 2-hydroxy species versus this 12-hydroxy species, which is on the path to the reactive metabolite formation, you'll see that 3A7 makes a lot more of the 12-hydroxy than the 2-hydroxy, whereas 3A4, the adult isoform, makes a lot less. So it's closer to 1-to-1 with the adult isoform. Why is this important? Well, if you go back to the schematic I was showing you before, where you have that unstable intermediate, which has got to recombine then to produce this innocuous species, either the 12-hydroxy or the quinone methide species, it suggests that metabolism with 3A7 is going through this pathway much more so than producing the 2-hydroxy species. So because we're making more of that 12-hydroxy, we can infer we're probably going to be making more of the quinone methide species as well. And in fact, we did the reaction phenotyping to show that the reactive species is mainly produced by CYP3A7 in neonatal human liver microsomes, similar, analogous to what we see in adult human liver microsomes where it's produced by CYP3A4, although there's a little bit produced by CYP2D6 as well. So how do we know that, potentially we're making more of this reactive metabolite with 3A7 than with 3A4? We decided to do an experiment where we had labeled S35 labeled glutathione, S35 labeled GSH. We did an incubation and then just basically assayed over time, how much of the that radio-labeled GSH is ending up in that reactive metabolite. And you can see with 3A4, over time, you start to plateau. This kind of makes sense, right? Because you're producing this reactive metabolite, which is adducting the enzyme and inactivating it over time. So eventually you're going to plateau because its production of that reactive metabolite's rate limiting because it causes inactivation of the enzyme that's producing it, right? In comparison with CYP3A7, although there's a lot less of that reactive metabolite produced because it's just less efficient at turning over the drug substrate, over time, you don't see that plateau, at least over the 60 minutes that we did this assay. So why would that be? Why would you see it? We understand why it's self-limiting with 3A4, but why isn't it self-limiting with CYP3A7? It turns out there's a structural answer to this. So what I'm showing you here is a crystal structure schematic of CYP3A4. And with CYP3A4, you can see the heme is in the active site here, the iron is in a rust color orange. I'm not showing you a substrate in the active site, but what I want to direct your attention to is a cysteine 239 residue, which kind of sits above the active site in this very dynamic region of the protein. And several previous studies have shown that when reactive metabolites are produced in that active site, these electrophilic metabolites, they end up adducting that cysteine 239 residue, and that leads to an activation of the enzyme. So it's almost like it's a sensor there, so that when you have these reactive metabolites being produced, they adduct that sensor, it activates the enzyme, so you're limiting the amount of reactive metabolite formation, toxic reactive metabolite formation that can occur over time. And in fact, when we do incubations with CYP3A4, remember that's the adult isoform, when we do incubations with that in the presence of nevirapine, and then we do whole protein mass spec, we can see a mass shift of 265 Daltons, which corresponds exactly to what you would predict for an adducted 3A4 species. And we further were able to characterize that by doing peptide MS to show that the adduct was located indeed at that on that peptide that contains that cysteine residue. So suggesting again that even in this case, when you're producing this reactive metabolite from nevirapine, it's inactivating the enzyme by adducting the cysteine residue. Now, what's interesting about this? So what's interesting about it is CYP3A7 doesn't have a cysteine residue in that position. And again, so we see increased formation of the reactive metabolite over time. It's not rate limiting because we don't see mechanism-based inactivation of the enzyme. And I have that data. I'm not presenting it today, but we did the same time course with incubation with nevirapine and CYP3A7, and we didn't see mechanism-based inactivation of it like we do with CYP3A4, suggesting that because you don't have that cysteine residue there that can be adducted, you continue to generate the reactive metabolite over time. So this is just showing you the proposed mechanism we have for this reactive metabolite generation, and then eventually how you, through rearrangement, how you have the quinone methide species, that electrophilic species adducting the cysteine residue on the 3A4 active site. All right, so the nevirapine story so far goes something like this. Nevirapine is metabolized both by CYP3A4 in the adult and by neonatal hepatic CYP3A7. We were the first to show that. And like CYP3A4, CYP3A7 also produces this reactive quinone methide metabolite. However, CYP3A7 is more resistant to inactivation than CYP3A4 because it doesn't have that cysteine residue. There's a serine in that position instead of cysteine. That allows it to produce more of this chemically reactive quinone methide species over time, leading to potential for increased toxicity in neonates. So why is this important? Well, it means that physicians need to consider other options potentially for neonatal HIV therapy than nevirapine. And right now we're taking this a little bit farther, and we're trying to look at some nevirapine analogs that might not get activated like this, like the parent compound does. And in fact, what we've done is we have an analog that's deuterated in that position, at 12 position. So you're less likely to generate that reactive metabolite. We've shown that the deuterated analog has the same IC50 against HIV reverse transcriptase, but it produces less of this reactive metabolite and doesn't inactivate the enzyme like the parent nevirapine does. So potentially we could develop a safer drug that way. Okay, so the second story I wanted to tell you about was CYP3A7 and its interaction with HIV protease inhibitors. So HIV protease inhibitors, when they first were produced in the early 90s, were really miracle drugs. Before this, if you guys have seen some of those movies, it was Dallas Buyers Club and things like that, you know, they talk about AZT and some of these reverse transcriptase inhibitors and other inhibitors that were around at the time, antiviral agents, which weren't very effective at all until the protease inhibitors came along. And they were really a lifeline to people that were infected with HIV. And because of that, we have a whole series of different protease inhibitors that are on the market now. They're great drugs. It turns out, though, they turned out to be really good CYP3A4 inhibitors, which is kind of strange because they're also cleared very rapidly by CYP3A4. And in fact, what they do is they induce production of the enzyme in the liver. So the drug you have around is inhibiting metabolism of other drugs, while at the same time getting it itself metabolized more quickly and leading to less efficacy and more viral resistant mutants. So because of this, people noticed early on that one of these drugs, ritonavir, actually works better as a CYP3A4 inhibitor than it does a protease inhibitor. So they used this to exploit the metabolism and slow it down so that they would get better PK with the other protease inhibitors. So right now, ritonavir is included in the cocktail, not for its protease inhibitor abilities, but for its ability to inhibit metabolism by CYP3A4 and CYP3A5. So why is that important, you know, for neonates, if you're going to give these drugs to neonates? So great, you're inhibiting CYP3A metabolism, so you're slowing down clearance of the drug, which may or may not be important depending on whether you're able to reach therapeutic levels, right? But you got to remember, CYP3A7 performs essential functions in the developing fetus and neonate. CYP3A4, again, is this garbage enzyme in the adult. If you inhibit it, you're not really going to be changing much in terms of homeostatic mechanisms. But in the developing fetus and the neonate, if you inhibit CYP3A7, that's going to lead to less production of these steroids downstream, particularly estriol. And remember, inhibition or lower levels of estriol lead to premature birth and low birth weight. So we decided to look at many of these HIV protease inhibitors, since we knew there were CYP3A4 inhibitors, to see if they were CYP3A7 inhibitors. And indeed, many of them did inhibit oxidation of this luciferin substrate. So this is a luminescent substrate. It's not anything, a native substrate of CYP3A7 or anything, but it just gave us an indication of how potent the inhibition might be with some of these protease inhibitors. And most of them are in the micromolar range, although one, nelfinavir, which isn't prescribed to neonates in general, is submicromolar. We also looked at ritonavir and in next generation, so these are called pharmacoenhancer drugs because basically they're just affecting the PK of other drugs by inhibiting metabolism. So ritonavir was the first generation pharmacoenhancer, but then later on they decided to modify it because it had this off-target protease inhibition, this weak protease inhibition, which led to production of viral mutants. So cobiosistat was the second generation drug that they made, and they basically engineered out the protease inhibition. So it's strictly a CYP3A inhibitor. And you can see with incubation with CYP3A7, cobiosistat has an IC50 of 0.32 micromolar. But ritonavir has an IC50 of 0.08 micromolar, so 80 nanomole, a very tight inhibitor. In fact, it's tighter than any of the other inhibitors that we looked at. About this time, we came across some papers that talked about the use of ritonavir and lopinavir combination therapy, also known as Calatra, in infants, neonates and infants that had been diagnosed with HIV. And also as preterm treatment. So they were giving it to the pregnant mother, and then once the infant was born, they were getting it as well. And what they found was there was a significant association between prematurity and this ritonavir boosted protease inhibitor therapy. So what do we mean by boosted? That means ritonavir is in there plus the protease inhibitor, compared with non-boosted therapy. Also, complications including preeclampsia, gestational diabetes, and liver toxicity were associated with premature birth. Another group looking at use of these drugs in this population noted that postnatal treatment with lopinavir and ritonavir of newborns from HIV-infected mothers also receiving the lopinavir-ritonavir combination was associated with transient adrenal dysfunction in the newborn, and a 19-fold increase of DHEAS concentration was observed in these newborns. So that really got us to thinking, wow, it sounds like CYP3A7 inhibition here, right? We showed that it's a potent inhibitor in vitro, but if you're inhibiting CYP3A7 in vivo, you would totally expect you're going to back up concentrations of that substrate, right? Because it's not getting cleared. And that's exactly what they see. They see sky-high concentrations of DHEAS. They see lower levels of the other downstream steroids, including estriol, and premature birth and low birth weight. So totally consistent with CYP3A7 inhibition. So our hypothesis here was that inhibition of CYP3A7 by ritonavir causes adrenal insufficiency and also may lead to dangerous drug-hormone interactions in the HIV-infected neonate. And the significance, which I'm going to show you at the end, is that the use of ritonavir as a pharmacoenhancer in neonatal HIV therapy may actually lead to poorer outcomes than treatments without ritonavir. So just with the protease inhibitor alone, in this case, lopinavir. And I'll show you why that is in a moment. But first, we needed a more relevant assay. The data I showed you earlier with the IC50s was with this luminescent fluorescent substrate, and that's not very reflective of DHEAS oxidation. So we went back to mass spec, developed a DHEAS assay, and we were able to get the kinetics for oxidation with purified CYP3A7. So we showed the KMs around 5 micromolar and the Vmax around 9. So keep that in the back of your mind. So then we did our IC50 studies again with lopinavir and ritonavir. In this case, lopinavir has an IC50 of around 6 micromolar. So that's close to the KM, even a little higher than the KM. So you wouldn't expect it impacting DHEAS oxidation that much. However, ritonavir has an IC50 of 50 nanomolar. So this is even tighter, more potent inhibitor of DHEAS oxidation than it was of the luciferin substrate. And in fact, we showed that ritonavir is a time-dependent inactivator of CYP3A7. So again, I'm showing you here on the left, the metabolism of secondary substrate as a function of time. So this is the percent enzyme activity remaining as a function of time and concentration of ritonavir. And as you increase ritonavir and increase the time, you see more inhibition of the enzyme, suggesting that it's a time-dependent inhibitor. And when we fit that to our hyperbolic curve, we get a Ki of 0.37 micromolar and K inact of 0.11 per minute. So a lot of times people will look at the K inact to Ki ratio to compare potency of inhibition. And in fact, when we look at that for 3A7, we retrieve a value of 320 per minute versus 3,200 per minute for CYP3A4. So CYP3, it's a more potent inhibitor of CYP3A4 even than CYP3A7, which is no surprise because it was developed initially to inhibit CYP3A4, or discovered initially to inhibit CYP3A4, not CYP3A7. Probably what's more important, though, is when we look at intrinsic clearance of the drug. So keep in mind, the whole point of having ritonavir here is that without ritonavir in the adult, lopinavir is getting cleared too quickly. So lopinavir is the active drug that's inhibiting the HIV protease, right? So it gets cleared too fast to be effective if you don't have this pharmacoenhancer there to inhibit 3A4 activity. And if you look at the right here, what you see in this graph, so what we're looking at is clearance of the drug in human liver microsomes. And the gray the gray line represents clearance in adult human liver microsomes in the absence of ritonavir. So the drug lopinavir gets cleared very quickly when you don't have ritonavir there. In neonatal human liver microsomes, it gets cleared much slower. So that's the black line and triangles up here. Now compare that when you have ritonavir in there in both cases, well, then you slow down metabolism by 3A4, but it doesn't seem to really affect metabolism by CYP3A7 because CYP3A7, remember, is clearing this substrate much slower than CYP3A4. So you don't really potentially even need ritonavir there. So this really leads us to the question, is ritonavir necessary in pediatric HIV treatment to inhibit metabolism? What are the pros for that argument? Well, it could keep lopinavir concentrations constant in spite of inter-individual variability. So people have mutations in these CYPs that may lead to faster or slower clearance. Now, not so much with 3A7, it's pretty much genetically homogeneous. We all have the same 3A7 here. But the other enzymes that are clearing it might be clearing it faster in some individuals and slower than others. What are the cons to having ritonavir there? Well, inhibition, as I've shown you, inhibition can lead to adrenal insufficiency, low birth weight, and dangerous drug-drug interactions. Because if we have mechanism-based inactivation of CYP3A7, these kids in the NICU that are on all these other drugs that are going to supposedly be getting cleared by CYP3A, now all of a sudden they're not getting cleared, so they could potentially build up to toxic concentrations. But probably more importantly, ritonavir may not be necessary at all to keep these lopinavir concentrations constant, like I just showed you with the clearance data. Whether you have ritonavir there or not, you seem to have about the same clearance rate in the neonatal liver. Okay, so I think I'll wrap it up there. Overall conclusions: nevirapine production of this reactive quinone methide metabolite by CYP3A7 could lead to increased toxicity due to lack of mechanism-based inactivation with this 3A isoform. So it's rate limiting in the adult because you have that reactive cysteine residue that acts as a sink for the reactive metabolites. But you don't have that. You have the serine residue in CYP3A7, so you don't have inactivation. So over time, and keep in mind, if you're on an antiviral, you're on it for your life, right? I mean, and so you have to maintain these high concentrations. So over time, you could be at increased risk of liver toxicity in the neonate. With ritonavir, this pharmacoenhancer agent that's just added in the cocktail to inhibit CYP3A, we found that it's a potent inhibitor of the CYP3A7-mediated oxidation of DHEAS, potentially leading to premature birth and adrenal insufficiency. However, ritonavir may not be necessary to treat HIV in the developing infant due to the slower clearance rate of lopinavir, the active drug, by CYP3A7. And if you've only taken one thing home with you today after this talk, I hope that its drug metabolism and disposition is different in developing infants than it is in adults. And you can't just extrapolate from the adult in all cases and expect drug clearance to be the same in the infant. So, of course, none of this happens without support, and I'm very grateful, particularly the support we got from the NIH, both NIGMS, and my grant just recently got transferred to NIAID, and they've been very generous in their support. And of course, the people in the lab who actually do the work, Sylvie Candle, she's a senior scientist in my lab with a lot of mass spec experience, metabolite ID. Dr. Brian Tooker, I think I saw him there somewhere in the little boxes. He's a postdoc in my lab. Hannah Work is our PhD student. And of course, Emily Hicks, our PRA. We also have collaborators working on that CYP3A7 structure in Michigan, Emily Scott, and a local collaborator who did some of the early metabolism work that I showed you over at Pfizer, Dr. Brian Baer. And with that, I'll take any questions. Maybe I don't know if I should stop. Should I stop share, Kevin, or?
Yes, you can if you'd like.
Okay. All right.
Well, thank you very much, Jed. I open the floor to any questions we might have.
Thank you all for the virtual claps. The animated claps. Steve, thanks, man. You got some skis behind you there. It looks like you're about ready to go skiing.
Yeah, those are some old Volants, if you're old enough to remember those things.
I do remember them. Yeah. Antiques.
Could I ask a question?
Yes, please.
Thank you. That's a really wonderful talk. I came a little late. You might have mentioned this. Among all
no worries.
Of the A4, are there any that are selective for A4 over A7, which might be something that could give you the enhancer function without affecting the neonate?
I'm sorry, I missed the first part of your question. Can you repeat it?
It sounds like what you need is ritonavir that is not very active towards A7, but is strongly an enhancer of A4, strongly inhibitory towards A4. And among all these, you showed us a lot of molecules that look like they're CYPA3 inhibitors. Did they show selectivity, differential selectivity for the two isoforms?
We're looking at that right now. So that's one of the things Hannah's doing in the lab. She has a really nice high throughput assay that she's developed, and which we're going to publish soon, hopefully. And she's screening a library of these compounds as we speak to see if we see any differences in selectivity between CYP3A7 and CYP3A4. I suspect there will be. We will see differences, significant differences in inhibition. I mean, there's still going to be, inhibitors, but it's just a question of, potency, right? So yeah, I mean, you could imagine. So it turns out in infants, they don't really have too much 3A4 for the most part. So we don't have to worry about that too much. So you can probably, I mean, if we believe our in vitro clearance data, you could probably get by with not having ritonavir in the cocktail at all. Just dose them with lopinavir. That would be my argument, because the fewer drugs you can give a baby, the better, right? But if you have an infant, let's say we're in the age where we can do genotyping and phenotyping, and we say, this baby is expressing some 3A4, so we do need to inhibit 3A4, but we don't want to inhibit 3A7, then I agree. Then it would be nice to have a selective inhibitor. So there's potential for developing that.
Because I think in your first part, you mentioned that the cysteine is selective for A4, and so that might be something you can take advantage of, to have this mechanism-based inhibition
Yeah.
that's selective.
Yeah. Yeah. So yeah, it looks like that cysteine residue has really evolved to trap these reactive metabolites that are produced by the enzyme. So I called it the garbage enzyme, but it's probably like the garbage man in your street, right? He picks up the trash about half the time. Half the time he dumps it all over the driveway, and you got to pick it up, you know? And so that's 3A4, right? I mean, a lot of times it's doing a very good job clearing drugs out of your system, or xenobiotics, you know, cancer-causing agents, whatever. But every once in a while, it makes something that's actually worse than the parent compound was, you know, more reactive than the parent compound. The classic example, again, is acetaminophen, right? It works great for your headache, but you take too much of it and you get liver tox, because these enzymes are making these reactive metabolites. So they're not very smart about how they eliminate garbage from your system. And so I think, at least the idea is out there, that's why nature has evolved this cysteine residue. You know, it sits right over the active site of CYP3A4, so that if you are making reactive metabolites, these very electrophilic species, that they're going to get trapped there and inactivate the enzyme, and so inhibit their own production. And that's what we see with nevirapine. So it's interesting that it seems like it's a mechanism that's evolved to deal with this, because you know, these enzymes are highly promiscuous, so they don't know which drugs are going to come on the market next year, right? But they have to be prepared to metabolize them. So how do they do that? Well, they have this big old active site that can let anything in there and produce all these different metabolites, most of which are innocuous, but occasionally, you know, they're going to produce something bad. So if you can limit the production of those reactive metabolites, it benefits you. But you don't see that with 3A7, so maybe it makes sense, because 3A7 is mainly expressed in the womb, and so, you know, presumably your mom is clearing all the bad stuff at that point. But yeah, sorry, I went on a bit there. But any other questions? Or?
I have a question. I'm Laura Brown. I'm at the University of Colorado. I'm a neonatologist.
Awesome.
I have
I need to meet more neonatologists.
Yes, and I have a colleague who is very interested in the CYP2s and their developmental regulation as it relates to APAP metabolism. Anyway, I've already sent him your name. But
awesome.
I have two questions. One, and I'm sorry if I missed this, but is the CYP3A7, so it sounds like it's developmentally regulated. So at some point,
that's right.
At some point, the expression decreases. And do we know when that happens or how that happens?
Well, you know, let me, here, if I can share screen just real quick again, if you guys will indulge me here. Just because I wanted to go back to one of those figures I showed at the beginning. Get all the way back here. Oops. Okay, this one here. Whoops. Okay. So yeah, it does look like it's developmentally regulated. What I'm showing you here is a panel from a group of livers. So there's been about 12 livers that have been pooled together. So and then they did a Western blot for the different 3A isoforms. So you can see, you know, before birth, the EGA is before birth, and then even out to 3 to 6 months after birth, most people are still expressing quite a bit of CYP3A7. There's a lot of inter-individual variability. So some people don't express as much as others. And you see this switch happens where you start, the amount of 3A7 goes down and the amount of 3A4 starts to go up. But it depends on the individual. Sometimes it happens earlier for some people or later for others. We don't really know what's controlling that. And in the fact that it happens before birth suggests that there may be some imprinting going on. It may not be, birth may not be triggering it, you know, the elimination of, or the switch from 3A7 to 3A4 per se. But What controls it? I don't know. That's really curious. And it's an essential role. I mean, you cannot, this gene is embryonic lethal if you knock it out. I mean, everybody has 3A7. So you have to have it to oxidize DHEAS and retinoic acid, at least before you're born, and for a little bit of time after you're born too, it looks like in most people.
Yeah, exactly. So thanks, that's super helpful. And I mean, I asked because obviously mothers are getting some of these drugs, but also we prophylax neonates, and a lot of them until we know that they're HIV negative. So while the I suppose the steroid metabolism is particularly relevant for the fetus, but I was also wondering if this also applies to the neonate, especially if it results in adrenal insufficiency, that could be problematic as well.
Exactly. And it does, you know. In fact, what I didn't show you is when they took the drug cocktail away, you know, then they recovered, and the adrenal insufficiency went away at that point.
Wow.
But these kids, they followed them up later in life, the ones that were negative, you know, and they still had problems even later in life. So it just kind of underscores, you know, how important the role this enzyme plays in the neonate and the fetus and the developing neonate.
Yeah, great talk. Thank you so much.
Thanks, Laura. Appreciate it.